How do scientists, actually peer inside a living thing, a whole organism, and track these tiny, intricate changes happening right there in its cells?
Uh-huh.
It's like wanting a window into life's, well, its inner workings, not just seeing the static parts, but watching them actually transform and adapt.
Right.
So today we're doing a deep dive into this really groundbreaking scientific paper. It seems they've unlocked a powerful new way to observe these fundamental processes, especially metabolism,
Hmm.
using a maybe surprisingly familiar little organism.
That's exactly it.
Yeah.
Yeah, we're stepping into quantitative proteomics, specifically how they've used stable isotope labeling,
Mm.
really cleverly, I think, in the nematode C. elegans.
The little worm.
The little worm, yeah.
Heh.
And this isn't just, another minor tweak. It's about overcoming a pretty significant hurdle.
Hmm.
It offers a genuinely novel technique. And that has, well, big implications for how we understand life at its most basic levels.
Okay, so let's unpack that.
Yeah.
Quantitative proteomics. What exactly are we looking at there, and why is tracking protein levels so crucial?
Right. So at its core, it's the study of how the amounts of different proteins change within a biological system. Proteins are, the actual workhorses in the cell.
Right, they do everything.
Pretty much. They dictate growth,
Yeah.
proliferation, how cells differentiate, develop, even how they survive. So if you can see how their numbers fluctuate, you get this incredibly valuable insight into what's really going on inside.
Makes sense. And I know there are different ways to measure these changes. What's the main strategy people use now?
Yeah, there are a few ways. You've got things like chemical labeling, where you sort of stick tags onto proteins after they're made.
Okay.
But metabolic labeling has really become, I'd say, the dominant strategy. That's where you build the tags into the proteins while they're being synthesized, using slightly heavier versions, isotopes of common building blocks.
Mm-hmm.
And the gold standard there is really SILAC-stable isotope labeling by amino acids in cell culture. It's super precise.
Why is it more precise than the chemical tags? Well, with SILAC, you're essentially barcoding every protein as it's made by feeding the cells specific amino acids that are just slightly heavier, like lysine with heavier carbon and nitrogen atoms.
Okay.
So every peptide fragment containing that amino acid has a very specific known mass difference compared to the normal light version. That makes identifying them,
Right.
and critically, quantifying the ratio between heavy and light much more straightforward and reliable than trying to make sure chemical tags react evenly with everything later on.
Right. And SILAC's been used successfully in lots of systems, hasn't it? Yeast, fruit flies, plants, mice.
Absolutely. It's incredibly powerful.
So what was the specific difficulty then in applying it to C. elegans? Why was that a hurdle? It's a common model organism.
That's a great question, and it really gets to the heart of what this paper solved. C. elegans aren't just cells floating in a dish, they're whole, complex, living animals.
Getting them to efficiently incorporate a specific labeled amino acid, not just in a few cells, but like throughout their entire proteome, and doing that without messing up their normal development or biology, that's been the real technical challenge. This paper basically cracked that code for C. elegans.
That's fascinating. So what was their core idea? How did they propose getting the worm to essentially label itself?
It was quite clever, really. Their central hypothesis was, what if we feed C. elegans a very specific type of bacteria, one that's an auxotroph, meaning it can't make a particular essential nutrient itself, it needs it supplied.
Okay.
So if the bacteria needs, say, the amino acid lysine, and we grow those bacteria in a medium where the only lysine available is our special heavy labeled lysine.
Then the bacteria become heavy.
Exactly. And then the worms eat those heavy bacteria. The idea was the worms would then have to use that heavy lysine from the bacteria to build their own proteins.
And you could track that heavy label in the worm's proteins using mass spectrometry.
Precisely. You create this custom labeled diet that builds the tags right into the worm's cellular machinery.
And they chose lysine for this? I thought SILAC usually uses both lysine and arginine for, like, maximum coverage.
Yeah.
Why just lysine here?
That's a sharp point. Yeah, using both usually gives you better proteome coverage because almost all proteins have at least one or the other. But they mentioned that trying to create a stable E. coli strain that needed both lysine and arginine externally proved tricky.
Okay.
Plus, there's a potential issue with arginine. In some organisms, it could be metabolically converted into another amino acid, proline, and that conversion can kind of mess with the quantification, making things less precise.
I see. So sticking with just lysine was maybe simpler and cleaner.
Exactly. They prioritized efficiency and the precision of the measurement by using only lysine.
Okay, so walk me through this ingenious feeding strategy. How did it work in practice?
It was actually elegantly simple in its setup. They took a known E. coli strain, ET505, which can't make its own lysine, the auxotroph. They grew batches of it in a defined liquid medium. The key was that this medium was supplemented with either normal, light lysine, they called it lysero, or the heavy isotope version, lys-8, which has 6 heavier carbons and 2 heavier nitrogens.
Okay, so 2 types of bacteria food.
Correct. Then they took these bacteria and used them to seed standard agar plates like you normally grow worms on. Then they just put young, unlabeled C. elegans, larvae the standard N2 strain, at day 1 onto these plates, and they let them grow and feed for 2 or 3 days until they reached an early adult stage.
And crucially, did the label affect the worms?
That's the critical part, no. They observed absolutely no difference in development, behavior, appearance, nothing between the worms grown on the light lysine bacteria versus the heavy lysine bacteria.
That's huge. It means the labeling itself wasn't interfering with the biology they wanted to study?
Exactly. It validated that the labeling method itself was benign.
So the worms looked normal, but how did they actually measure how much heavy lysine got incorporated? What was the readout?
They used a technique called liquid chromatography, tandem mass spectrometry, or LC-MSMS. It's a super-sensitive way to measure the mass of molecules.
Right.
First, they basically homogenized the worms to get all the proteins out. Then, they used a specific enzyme, lysyl endopeptidase.
Okay, what does that do?
It's like a molecular scissor that cuts proteins only after a lysine residue.
Ah, so that guarantees most of the resulting pieces, the peptides, will contain the labeled lysine if it was incorporated.
Precisely. Very important detail. Then the LC-MSMS system separates these peptides and weighs them with incredible accuracy, allowing them to clearly distinguish between the peptides containing the light lysine and the heavy lysine.
And what did those measurements show? Did the worms actually incorporate much of the heavy lysine?
The results were really quite impressive. They found near complete incorporation. After just 1 generation feeding on the heavy bacteria, reaching the L4 larval stage, the worms had incorporated 94.6% heavy lysine, plus or minus about 2.8%.
Wow, that's high.
It is. And if they let them grow just 1 more day into early adulthood, it went up even further to 97.5%, plus or minus 2.2%. Really efficient labeling.
And accurate, too.
Very accurate. The standard deviation they reported was less than 0.22. What that means in practice is that for about 2/3 of the protein ratios they measured, the value was extremely close. Less than a log 2.22-fold change from the expected 1.1 ratio when they mix light and heavy samples equally. That's the kind of precision you need.
So just to help visualize this, their figure 1 basically shows this concept. You have 1 batch of worms grown on light food, another on heavy food. They look identical. You mix them together, say exactly 1-to-1. Then you extract the proteins, chop them up with a lys, and run them through the mass spec.
Right.
And for any given protein, you'll see pairs of peaks in the data, one for the light peptide, one for the heavy peptide, separated by exactly 8 daltons, which is the mass difference of that heavy lysine. And the ratio of the heights or intensities of those peaks tells you the relative abundance of that protein in the original light versus heavy populations. If they're equal height, it was 1.1. If one is twice as high, it was 2.1, and so on.
That's the core principle. It's really elegant.
So, okay, they've nailed down this precise labeling method in the whole worm, but then they took it a step further, which sounds really powerful. They combined it with RNA interference, RNAi?
Yes. That's where it gets really exciting from a functional genomics perspective. RNAi is this fantastic tool, especially in C. elegans, for basically switching off or knocking down specific genes to see what they do.
So how did they merge these 2 techniques? Did they have to modify the bacteria again?
They did. They needed to make their lysine-requiring E. coli strain, ET505, also capable of inducing RNAi in the worms that ate it.
Right, because normally you use specific RNAi feeding strains of bacteria.
Exactly. So they made 2 key genetic changes to ET505. First, they integrated something called a DE3 construct into its genome. This makes the bacteria produce an enzyme, T7 polymerase.
And why is that needed?
T7 polymerase is required to actually produce the double-stranded RNA, dsRNA, from plasmids that you introduce into the bacteria. The dsRNA is what triggers RNAi in the worm.
Okay, step 1. What was step 2?
Step 2 was deleting the gene for an enzyme called RNAi-3. RNAi-3 normally chews up double-stranded RNA in bacteria.
Ah, so removing it protects the dsRNA needed for the RNAi effect?
Precisely. You want lots of intact dsRNA to be delivered to the worm. So they engineered this new strain, deleting RNAi-3 and adding the T7 polymerase capability, and they called this modified strain NJF01.
Okay, NJF01. Did modifying the bacteria like this cause any problems? Did it affect the labeling?
They noted it grew a little bit slower than the original ET505 strain, but importantly, it was still a perfect lysine auxotroph. It still needed lysine provided.
Uh-huh.
And crucially, when they tested it, the amount of heavy lysine incorporation in worms fed on NJF01 was virtually identical to worms fed on the original ET505.
So no compromise on the labeling efficiency. That's great.
Huge win, yeah. The modifications needed for RNAi didn't mess up the labeling part.
And did they check if NJS01 actually worked for RNAi?
Oh, yeah. They validated it thoroughly. They showed it could effectively knock down known genes, like they targeted DORDO13.
The jumpy gene.
The dumpy gene, yeah. And sure enough, the worms fed NJF01 expressing DOPI13 dsRNA showed that characteristic short, fat, dumpy phenotype.
Okay.
They also tested it in worms engineered to express a fluorescent protein fusion, GFP, attached to RB7. They fed these worms NJS01 expressing GFP dsRNA.
And the GFP signal went down.
Exactly. They confirmed it both by looking under a fluorescence microscope, less green signal, and by Western blotting, which biochemical measures the protein levels. Both showed efficient knockdown.
So NJS01 worked just as well as standard RNAi bacteria.
Yep, the validation was solid. It was ready for the combined experiment.
All right, so the moment of truth, combining heavy labeling and RNAi knockdown. How did they set that experiment up?
Okay, so they took those transgenic worms, the ones expressing the GFP-RB7 fusion protein. They fed one batch of these worms on their new NJF01 bacteria that were labeled with heavy lysine. Lysine, indeed, were expressing the GFP dsRNA to knock down GFP.
So heavy worms, GFP gets knocked down. Got it.
Then they took a second batch of the same transgenic worms and fed them on NJF01 bacteria labeled with light lysine, lys0, which
Right.
carrying just an empty RNAi vector one that doesn't target any gene. So these are the control worms.
Light worms, GFP is expressed normally.
Okay. Then they mixed these two populations of worms together, 1-to-1, the heavy GFP knockdown worms and the light control worms.
And ran the mass spec analysis on the mixture. What did they see?
The results were crystal clear, just as they'd hoped. When they looked at peptides specifically from the GFP protein or the RB7 protein it was fused to, they essentially only saw the light versions.
Because the GFP was knocked down, reduced in the heavy worms.
Exactly. But when they looked at peptides from many other unrelated proteins that shouldn't be affected by the GFP RNAi, they saw both the heavy and light versions present in roughly a 1-to-1 ratio.
Wow, that perfectly demonstrates it worked. They successfully combined the heavy lysine labeling with targeted RNAi knockdown in the same population.
It's a massive step. It means you can now directly measure at the protein level across the whole proteome the specific consequences of silencing almost any gene you choose in this whole living animal. It opens the door to mapping entire protein network responses.
That is incredibly powerful. So, having established this technique, they then applied it to explore some real biology, right?
Right?
They looked at this factor called NHR49.
Yes, NHR49. It's a nuclear hormone receptor in C. elegans. Think of it like a master switch, and it was already known to play a key role in controlling genes involved in lipid metabolism, how the worm processes fats.
Okay, so a known fat regulator. How did they use their new technique to study it?
They basically did two parallel experiments to see what happened to the worm's proteins when NHR49 function is messed with.
Okay.
First, they did a functional loss experiment. They used worms that were actual mutants for the NHR49 gene. The gene was broken. They compared these mutants to normal wild-type worms. They used that label switch design again, feeding some mutants light lysine, some heavy, and doing the opposite for the controls, then mixing. This helps cancel out any subtle variations. From this, they identified almost 4,000 proteins and managed to reliably quantify about 2,900 of them. And they found 143 proteins whose levels were significantly changed up or down in the mutants lacking NHR49.
Okay, 143 proteins affected in the mutant. What was the second experiment?
The second was an RNAi knockdown experiment. Here, they used their new RNAi-compatible bacteria, NJF01, to specifically silence the NHR49 gene in otherwise normal worms. They did this for 2 generations to ensure a strong effect.
Using the new combined technique.
Exactly. They ran this experiment in duplicate. This time, they identified even more proteins, over 4,600, and quantified about 3,470. In this RNAi setup, they found 330 proteins were significantly up or down regulated when NHR49 was knocked down.
330 in the RNAi, 143 in the mutant. Why the difference in numbers?
Well, RNAi knockdown isn't always exactly the same as a complete gene knockout or mutation. Sometimes the knockdown is incomplete, or it might have slightly different timing or off-target effects. But what was really important was the overlap.
Right. Did the two different methods, mutant versus RNAi, point to the same kinds of changes? Was there consistency?
Yes, and that was a key finding for validating the approach. They found a statistically significant positive correlation between the protein changes seen in the mutant worms and the changes seen in the RNAi knockdown worms.
So generally, proteins that went down in the mutant also tended to go down in the RNAi and vice versa.
Exactly. It showed the two independent methods were revealing a consistent biological signal related to NHR49 function. In fact, they found 99 specific proteins that were significantly regulated in both experiments.
99 proteins consistently affected. That really boosts confidence in the findings.
Absolutely. It shows the robustness of the results.
So what were these proteins that changed when NHR49 function was lost? Did it confirm its role in lipid metabolism?
It absolutely did, and in a really clear way.
Yeah.
They performed what's called gene ontology, or GIGO term, analysis on the list of regulated proteins. This basically asks, are proteins involved in particular biological processes overrepresented in this list? And they found a massive overrepresentation of downregulated proteins, proteins whose levels decreased that were involved in lipid metabolism.
So losing NHR49 function caused a drop in the levels of fat processing protein.
Precisely. They saw significant downregulation of key enzymes like fatty acid desaturases, things called FAT1, FAT2, FA5, FAT6, which are crucial for making certain types of fats.
Okay.
And also many other proteins known or predicted to be involved in breaking down fatty acids, transporting lipids, and general energy metabolism. It painted a really coherent picture at the protein level.
And did this protein level picture match what was known from studying gene expression, the mRNA levels related to NHR49?
Yes, another important connection. They specifically noted that their protein abundance changes correlated significantly with previously published data on mRNA levels for genes known to be regulated by NHR49.
So the changes are happening at the gene activity level and translating into changes in the actual amount of protein workers doing the job. That provides a much more complete understanding.
Exactly. It ties the whole regulatory pathway together, from the gene control by NHR49 right down to the functional protein output.
So what was the researchers' main takeaway message from all this work?
Their overarching conclusion was pretty clear. They argued that this combined approach, marrying quantitative proteomics using their labeling method with powerful genetic tools like RNAi and C. elegans, is going to be incredibly valuable. It will significantly push forward our understanding of how fundamental processes like cellular signaling and metabolism are coordinated within a living organism.
And bringing this back to you, our listener, why does this matter beyond the world of worm research?
Well, the way cells manage energy, build molecules, and process nutrients, metabolism is absolutely fundamental to all life, including ours.
Great.
And when these fundamental processes go wrong, when signaling and metabolism get out of sync, that's often at the heart of many human diseases. Think about diabetes, obesity, cardiovascular problems, many metabolic disorders. So developing tools like this, which allow us to dissect these pathways with such precision in a model organism like C. elegans, which shares many basic metabolic genes with us, is a really vital step. It helps us understand the basic rules of the game, which is essential for figuring out what goes wrong in human health and illness.
It's quite a journey we've covered then, from figuring out how to feed worms heavy amino acids to combining that with gene silencing and finally using it to map out the protein landscape controlled by a key metabolic regulator.
Yeah, it really showcases the ingenuity involved in developing these biological tools and how studying a seemingly simple organism can yield really profound insights.
It really is amazing how much we can learn from these tiny creatures. They hold so many clues.
They really do. It's a constant reminder that fundamental biology is often conserved, which leads to a sort of provocative thought, perhaps. Well, if we can now track these metabolic shifts so precisely at the protein level in C. elegans, what could that mean down the line for our ability to spot really subtle, maybe very early, signs of metabolic problems in more complex systems, maybe even in humans?
Like identifying biomarkers for metabolic disease risk much earlier than we can now.
Potentially. Could this kind of detailed proteomic insight eventually help us find those early warning signs, paving the way for maybe preventative strategies rather than just treating metabolic diseases after they're fully established?
That's a really powerful thought. Understanding these tools and what they reveal doesn't just advance basic science, it could genuinely change how we approach health and disease in the future. Truly fascinating stuff.